So good morning, and thank you very much for inviting me back. It's the first time that I have been invited back without having to apologise, so I'm very grateful to everyone. I have colleagues here today who are checking on me. I've been asked to speak about the methodology extension called Active Protective Biology, and some of the work on the clinical studies that we are doing with the Complex Disorders Alliance and the Metadora Institute. Not being entirely obedient, I will bookend that with a quick update on the reproduction of results that we have done on our analytical studies in myalgic encephalomyelitis and long COVID, and then talk a little bit about how we propose to bring a laboratory-developed test to market and how we might partner with the community to drive forward. So we start, I think, in quite a sobering position, despite all of the efforts of everyone in the room. We still don't know very much about these two diseases, and close on 500 million people around the world are living with these conditions, waiting impatiently for us to make progress. And the costs are substantial to the individuals, to healthcare, and to society. And it's disappointing that the biopharmaceutical industry are not more active in finding new medicines and treatments. And in part, it's because they don't know where to begin. We don't have clarity on the underlying mechanisms. We don't know much about which tissues are being effective. And we do not have absolute clarity of diagnostic tools that will allow effective care of patients beyond the specialist groups that we have represented today. And genetically, being able to link mechanism of disease with genetic evidence is regarded as the pharmaceutical industry's biggest driver of de-risking discovery. We don't have much known about the underpinnings of this disease, but I'm looking over to Professor Ponting. I believe this will change soon, and I'm going to applaud him when it does. In the meantime, Precision Life has brought to bear its own unique analytical approach to analysing genomic and multi-omic data. And last year, we talked about the 14 genes that we had identified in an analysis of myalgic encephalomyelitis, and the 73 genes through a long COVID analysis that we had done analysing a data set from Sanogenetics. Of those 14 genes in myalgic encephalomyelitis, 9 were in common with the long COVID data set, and that's a very interesting potential linkage that connects the two from a mechanistic potential. We've now been working towards reproducing those results, and I just want to remind you of how we think about analysing disease sets just before I talk about that. So last year, some of you will remember, I put up this colourful set of splodges describing how we get to a hypothesis-free, mechanistic stratification of patients. And we do this by looking for combinations of genetic features, SNPs, that occur in subgroups of patients but don't occur in cases. We call those combinations signatures, and then we cluster those signatures by the patients who they co-occur to build communities of signal. And then we do analysis. We do bioinformatic annotation analysis, and we link through to, because we know which patients are represented in each of those coloured clusters, we can link through to questionnaires or real-world evidence to see whether what we find from a hypothesis-free perspective is substantiated in the clinical notes. So the green group, just to remind you, is centred on a main gene called ACAP1. ACAP1 is involved in energy release in mitochondria, and this is the subgroup of people who with myalgic encephalomyelitis have post-exertional malaise as their primary symptom. So that's very encouraging, and we can step through this particular garden, if you like, of clusters. We have the blue group where we have SLC611 as a gene which encodes for a particular neurotransmitter. These groups of people cannot metabolize phenylalanine up to serotonin, and this group complain of brain fog as part of their disease. And we can go through a number of other routes. And so this was an encouraging start. We did a similar analysis in long COVID with the Sano Gold study. We divided that population into two subgroups based on phenotype. One who were reporting a very severe change in their health with long COVID, and another group who were reporting fatigue as one of their dominant symptoms. We analysed that, and now we have gained access to all of us with the support of the Complex Disorders Alliance, and sought to see whether those signatures that we saw in a UK population, predominantly white Caucasian, were reproduced in all of us. We have had some challenges with all of us. The QC criteria that the All of Us group have used on their genotyping means that there's missing data. So not all of the signatures and genes that we would like to be able to explore in all of us are available for testing. And we've also been very conscious of the fact that this is all new biology. And so we've taken a very cautious approach to making sure that the individuals in our cohort that we're looking for these reproduction of signals really meet the diagnostic criteria of long COVID. So of the 51 genes that we were able to test for in all of us, 45 replicate overall, and the signatures 1,766, 1,368 replicate. And I think one of the most encouraging points for us is that we saw good replication in diverse ancestries, something that is not guaranteed when we take some of the classical approaches to analysis. So that's a very positive start. 7 of those ME genes, 7 out of the 9 ME genes that linked long COVID, we could validate. The other 2 were not in the data set and so we couldn't test those. And of the 13 genes with potential drug repurposing candidates, they were reproduced, so a good start there. In ME, we have done some initial work using the first batch of data that we've had access to. The results are encouraging, around 65%. That will get better as we get the final data set to perfect the reproduction. So turning to active protective biology, our active protective biology approach recognizes that not everything in biology is driven by risk, right? We are all aware of people who age more gracefully, seem to have healthier lives, and clearly there is a difference between us. We're all exposed as we go through lives through potential risks associated with our genetics, with our environment, with comorbidities. But some of us seem to bear that well. And so we wanted to go out and actively look for these protective mechanisms. As I was explaining to Nancy Klimas over dinner last night, maybe it's not the sex that makes you pregnant, it's the lack of protection. And I've just got a $10 bet there, Nancy, for using that in my talk. And so, last year I talked about in myalgic encephalomyelitis, on the violin plots that we use, a substantial difference between the cases and the controls. And in thinking about how we might find a path to identify these active protective mechanisms, there's a clue. Because what are those people doing in the control population, healthy, who seem to have a large number of genetic risks? Because if biology was simple, and we all know it's not, they really should be cases, but they're not. And so we can use that insight to go looking in the UK Biobank for a non-ME/CFS population that has all of those risks but remain healthy. So we bring the results of our initial analysis to the table. We eliminate young people, not because we don't like them, we are jealous of young people, but they may not have had the environmental exposures to progress to a disease phenotype. So we look for people who are as old as possible, have had every opportunity to become sick, and who haven't. We ignore those who are low risk. The ones high risk and healthy are the protected cohort, and then we compare them. We compare our case population to the protected cohort to see if we can find genes that seem to be protecting individuals and stopping them getting sick. And we think this is a huge opportunity for new drug targets, potentially therapeutic mRNAs, and of course Precision Life works in a number of complex chronic diseases. And if there was a therapeutic vaccine that prevented people getting Alzheimer's, I would be at the front of the queue as I'm looking down the barrel of getting older. So the results of the ME/CFS active biology analysis, we did in fact find 430 unique SNPs in 239 signatures, those combinations of 2 or 3 or 4 or 5 combinations, and they clustered up into 12 communities. And we've published this work. There are a number of mechanisms. Personally, I was pleased to see a lot of tyrosine biology, given some of the comments from last year. But you can see there is a member of the tyrosine phosphatase family. Down here, there is an enzyme responsible for catabolism. Some changes in perhaps how patients are able to metabolize these amino acids may be why they're experiencing disease. Just to dig into that a little bit more, some of those genes actually gave evidence or further supported some of our hypotheses of what was driving risk. So CDC14A is a member of the cell division groups. It plays a key role in insulin secretion via AMP-activated protein kinase. That is something that occurs in skeletal muscle. It supports the involvement of ACAP1, clock, and some of the other genes I've talked about. We've got some calcium calmodulin-dependent protein kinase in the mix, where there's definitely been an association with diabetes risk. Interestingly, CAMK1G is more commonly expressed in neuronal tissue. It's definitely part of a stress response in combination with EBP4-1LAB. We've definitely seen that gene have increased methylation in as individuals experience stress response, and intriguingly, CAMK1G is also a gene that we see in our ALS studies. And in GAB4, we've got a potential linkage to inflammation and the development of autoimmunity. So knowing both what risk and protective biology, we now have a real opportunity to evaluate what an individual's potential disease risk looks like through a simple cheek swab, buccal swab, or if there's an existing DNA record, we can do a genotyping array. We can look for both disease and protective signals, and the overall risk for an individual should be the sum between the risk and the protective mechanisms. And this is the work that we are trying to push forward in clinical studies at the Metrodora Institute over in Salt Lake City in Utah, which is why you got me as a second-rate speaker last year instead of my excellent colleagues who were over in Salt Lake trying to initiate these studies. METX is their biobanking collection study. We have a sub-study within that called MELLOW, which is looking at ME and long COVID work. And as of yesterday, I believe 140 patients of data is heading over to Tempus to be genotyped, and we're making some progress. So at MELLOW, we're looking to recruit up to 1,000 patients. We will be doing surveys and genotyping studies on them. We will score our patients. We will return results to patients using clinicians, and we'll be looking to evaluate the diagnostic accuracy. And this means that we're starting to consider how do we support the clinical community with actionable information that actually can help them in caring for individuals, because this is a heterogeneous and complex disease. And so, the reports look like many of the precision oncology reports that some of you will have seen, except at the front there are these radar plots. Now, METX isn't necessarily recruiting people who are only have long COVID or ME/CFS. So not everyone that we've returned results to will have these diseases, but nevertheless, you're already seeing sort of patterns emerging. So patient 3 and 4 have a kind of what I call the north-south pattern. We also see some more east-west type individuals, and I think it more than anything, it calls out something that I often say on calls, is there's no patient who's polite enough to walk into a clinician's surgery with just one mechanism of action driving their disease. So, many of these patients will need more than one therapeutic to resolve their underlying condition. We've also been building out at Metrodora the infrastructure to be able to return results to individuals. And so the way the recruitment works is an individual will receive a QR code. It's essentially like shopping online. Hurrah, shop online for your health. And you can enroll. You have the opportunity to share your EHR record if you would like through the Care Evolution platform. There is a survey to complete. If you would like, if you meet the criteria and you would like to go forward, all kitting and sampling is done at Tempus. Kits are sent to the patient, returned and genotyped and QC'd by them in the wet lab. We then come in with a system that allows us to provide a dry lab interpretation layer. This is essentially software as a medical device, where we QC the response. And I think one of the things I'm just going to pause for and make the point is we talk a lot about, building greater understanding of our disease. We're building disease models, if you like. We talk about disease schema, but we then have to think about how we deploy those in the clinic. And so one of the advantages of the precision life approach is these are deterministic models. They can be deployed, and those will then drive an automatic reporting, a consistent reporting for patients. And then they get a clinical decision report, goes to the clinician, and those reports are shared with patients. Very little, small insight, encouraging insight, that's all I can say it is. But with some of the first individuals who went through the process, who were very engaged, they reported it was the first time they felt their symptoms were accurately described and they felt seen, if you like. And the clinicians said that this type of reporting was much more useful to them than the standard whole genome report. So I think that's very exciting. We have to proceed with a measure of caution. Everything here is novel biology that we're discovering, and we want to bring the biopharmaceutical industry in alongside us. And so we need more clinical studies to validate these mechanisms that we're seeing. We would like to enable the community to do more of focused repurposing, so to be able to select cohorts based on mechanisms that are likely to respond to repurposing candidates. It's the fastest way we know to benefit the patient community. And also it will provide evidence that these mechanisms are disease modifying, reasons for the industry to come in behind us and develop treatments, and also prove that we can select individuals who will respond to those mechanisms, de-risking investment in trials. We have our own process, very similar to the one described on finding potential repurposing targets. We've got many more than we can work with individually. We're very keen to collaborate with the community. I have 4 asks. I'm very aware I'm running over. So one is, I stood here a year ago and said we are data scientists first and disease experts a long way behind. There are groups here who have really strong knowledge. They've been reaching out to us, and we've been doing what's called a seeded analysis. Think of it as a fine mapping. If you have a hypothesis that a particular mechanism is involved, we can drive our systems to find that biology. We're looking for individuals who would like to gain access to that LDT to enable precision drug repurposing. And if anyone's got really good data who could help us make more progress, we would love to work with you. There's an amazing team of people at Precision Life behind this, and I'll leave you with the papers. Thank you very much.